Hello and welcome to this video covering recent developments in research exploring the connection between COVID-19 exposure and female fertility. My name is Andrew Ng and I'm currently enrolled in Middlebury College's Topics in Reproductive Medicine course. This presentation is catered to those with knowledge of the biology surrounding reproductive medicine. However, I have attached links in the video description to other resources that cover the biology underpinning the topics I will cover. Here's what we will cover in this video. First, a bit about COVID-19 and its underlying virus. Then, implications for the female fertility and specifically pregnancies with COVID-19. Severe acute respiratory syndrome coronavirus 2 refers to the virus itself, while COVID-19 refers to the disease that occurs due to the virus. It emerged in Wuhan, China during the end of 2019 and was officially declared a global pandemic by the World Health Organization on March 11, 2021. Since then, it has accounted for over 5.67 million deaths, and 1 in 5 Americans have contracted the virus. It is commonly known for its impact on lung function, resulting in respiratory failure and pneumonia, but it has also been commonly implicated in heart, liver, kidney, and nervous system complications. The virus is identified as a member of the beta coronavirus subgroup with a positive sense single-strand RNA located within a nucleocapsid contained in an envelope. The viral structure includes characteristic spike S proteins that are projected from the virion surface and assist in viral cell entry, membrane M and envelope E proteins that assist in viral assembly, and the N protein, which forms the nucleocapsid. Of specific interest is the S protein due to its role in cell entry and thus viral replication. This S protein is comprised of two unique subunits, S1 and S2. The S1 domain functions in virus binding to the host cell membrane, while the S2 domain is responsible for the fusion of virus to host cell membranes to facilitate the viral genome in entering the host cell. There are two specific host proteins that are implicated with the spike protein. The first is transmembrane protease serine 2. By itself, the SARS-CoV-2 S protein cannot properly attach to the host cell. Instead, it must first undergo priming by cellular proteases, which entails the S protein cleavage at the S1, S2, and S2' site, and allows fusion of viral and cellular membranes via the S2 unit. The transmembrane protease serine 2 is responsible for this priming in humans. The S protein is then prepared to engage with angiotensin converting enzyme 2 as the entry receptor. Thus, both TMPRSS2 and ACE2 are required for host invasion and viral spread. Looking a bit more at the function of ACE2, as the name suggests, it is capable of converting angiotensins. Specifically, it is a zinc metalloprotease with a high affinity for angiotensin 2 to generate angiotensin 1,7. Angiotensin 2 reacts with angiotensin 2 type 1 receptor, commonly denoted AT1R, and activates the system to induce vasoconstriction, promote cell proliferation, and maintain hydrosalinity balance, amongst many other functions. Angiotensin 1,7 induces antagonistic effects compared with AT1R, enhancing vasoconstriction, providing anti-inflammatory effects, and alleviating metabolic syndrome. SARS-CoV-2 downregulates ACE2, resulting in a lower conversion of angiotensin 2 to angiotensin 1,7. This disrupts the finely tuned system and is believed to be the fundamental cause of issues that proceed. Both these host proteins are expressed in cells all throughout the body, especially the lungs, hence the prevalence of lung-related complications. However, they have also been shown to be highly expressed in other organs, including but not limited to the heart, kidney, and liver. However, ACE2 and TMPRSS2 are also found in various areas of both male and female reproductive tracts as well. The remainder of this video will review the growing amount of literature surrounding SARS-CoV-2 and its implications on fertility in the female system. To begin, we will cover the female fertility as it relates to COVID-19 exposure. The system is generally considered to have 5 main parts: the ovaries, the fallopian tubes, the uterus, the cervix, and the vagina. The ovaries are the female gonads responsible for secreting hormones, protecting eggs that a woman is born with, and releasing eggs for fertilization. The fallopian tubes are conduits between the ovaries and the uterus in the female pelvis. They function as channels for oocyte transportation and fertilization. Once the egg has left the ovary, it can be fertilized and implant itself in the lining of the uterus. The main function of the uterus is to then nourish the developing fetus prior to birth. The possible implications of COVID-19 on female fertility was established quite quickly after the virus was initially identified. In fact, this specific article by Jing and colleagues was submitted only 10 days after the World Health Organization declared COVID-19 a global pandemic. Since its publication, this paper has served as a basis for much of the research surrounding female fertility and COVID-19 and has been cited by nearly 200 other authors exploring this topic. Their intuition was that cells that express the entry site for SARS-CoV-2, specifically the ACE2 receptor, could be at risk for virus-related complications. Reviewing previous literature in both animal and human models, as well as analyzing human genomic databases, researchers have identified that ACE2 is commonly present throughout the female reproductive system and thus may be a potential target for SARS-CoV-2. In the ovary, ACE2 is expressed in the stroma and granulosa cells and are highly expressed in the oocytes. As a reminder, ACE2 plays a key role in balancing the angiotensin system, specifically angiotensin 2 and angiotensin 1-7. These angiotensins are well researched in the reproductive system and hold important functions within the ovaries. Angiotensin 2 induces steroid secretion, facilitates follicle development and oocyte maturation, contributes to follicular atresia, influences ovulation, and maintains corpus luteum progression. Angiotensin 1-7 promotes the production of estradiol and progesterone, enhances ovulation and the resumption of meiosis in oocytes. Therefore, the proper functioning of ACE2 and keeping the correct balance of angiotensins is critical in the ovaries. ACE2 mRNA has also been identified in the uterus and vagina of humans, with claims that it is highly expressed in the epithelial cells in a phase-dependent manner. Specifically, higher expression in the secretory phase than in the proliferative phase. With that being said, there are relatively low levels of ACE2 transcripts in the endometrium, and the ACE2 protein itself is not found in the endometrium. Angiotensin 2 plays a dual role in the vascular bed and endometrium regeneration and initiates menstruation through spiral artery vasoconstriction. Specifically, angiotensin 2 increases the proliferation of uterus epithelial and stroma cells and enhances endometrial fibrosis, an effect that can be inhibited by angiotensin 1-7. Thus, the balance of angiotensin 2 and 1-7 is imperative for the proper menstrual cycle regulation and maintenance of the uterus. With the potential for SARS-CoV-2 infection established throughout the female reproductive tract, over the last 2 years, research has focused on the implications of COVID-19 exposure and fertility. Moving from looking at viral potential to looking at how fertility measures actually present themselves in women exposed to COVID-19, we will begin looking at reproductive hormones. This information comes from a study by Lee and colleagues and is one of the most cited studies looking at reproductive hormones as they relate to ovarian fertility. They compared the sex hormones of 91 women diagnosed with COVID-19 with those from age-matched controls. Hormones were measured from blood samples from the early follicular phase. The data showed no statistically significant difference in all of the sex hormone concentrations between the COVID-19 patients and the controls. This result indicated that the ovarian endocrine system of most female COVID-19 patients was not seriously affected. However, some patients had abnormal changes in their sex hormone concentrations, such as high concentrations of FSH and LH, which may indicate ovarian suppression in these patients. Previous research demonstrates that when placed under acute stress, ovarian function is usually suppressed to ensure normal operation of essential organs and anovulation has been reported in many acute diseases. While these hormones also play critical roles in other areas of the reproductive tract, less work has been done exploring those areas and is an area of future research. Staying within the ovaries, we will next look at ovarian reserves, a term that is used to determine the capacity of the ovary to provide egg cells that are capable of fertilization. Anti-Mullerian hormone, commonly referred to as AMH, is recognized as a key marker for ovarian reserves. AMH is produced by the granulosa cells of small growing follicles in the ovary. Thus, serum AMH levels have been shown to strongly correlate with the number of growing follicles. There have been numerous studies that have examined the serum AMH levels between women with and without COVID-19. 4 of the 5 most commonly cited studies I found on this topic, AMH levels did not vary between groups. These studies covered a collective of 469 women. Meanwhile, one study that I found lower levels of AMH when looking at a group of 78 individuals with COVID-19 versus those who did not. Overall, this looks to suggest that the ovarian reserves are not impacted. Along those same lines, other aspects explored are the presence of the virus in both oocytes and follicular fluid. The in vitro fertilization process provides the opportunity to closer research these areas, as egg retrieval provides the opportunity to provide more closely examined eggs for the presence of SARS-CoV-2. Generally, follicular fluid reflects oocyte quality, and alterations in its natural state can have detrimental effects on reproductive function. Multiple studies have examined these eggs and found that the virus is neither present in either the oocyte or the follicular fluid. However, in all instances, the antibody, anti-SARS-CoV-2 IgG, was identified in follicular fluid. Accompanying the antibody was lower levels of vascular endothelial growth factor, VEGF, and interleukin-1 beta. VEGF is known to influence the development of ovarian vasculature and is thus implicated in the nutrient supply to the follicle. Meanwhile, interleukin-1 beta is known to affect folliculogenesis and atresia, imperative aspects of proper egg development. Ultimately, these are 2 mechanisms by which exposure to the virus could be detrimental to fertility. Not directly related to the virus, but the body's immunological response to it could hinder oocyte quality. Moving from the analysis of oocyte and follicular fluid composition to the analysis of oocyte retrieval, a number of studies have compared the IVF egg retrievals and embryo developments of individuals who have had COVID-19 and those who have not. These studies can provide direct evidence for COVID-19's impact on assisted reproductive technologies, but can also be proxies for general female fertility. Here I summarize the main findings amongst 5 of the most prevalent studies. Bentov and colleagues took a look at women going through egg retrieval that were either vaccinated, COVID recovered, or unvaccinated, and measured the number of oocytes retrieved, oocyte yield, mature oocytes, and oocyte quality biomarkers, finding no differences between any groups. Wang and colleagues conducted their studies in the largest IVF center in Wuhan, looking at similar metrics but including fertilization rate and blastocyst formation rate. Their results indicated that the blastocyst formation was impeded in those who had recovered from COVID-19, while all other metrics were held the same. Harrow and colleagues found different results, with women who had had COVID having significantly lower number of retrieved and mature oocytes. Continuing on with the studies that looked more at qualities of embryos, we have Orvieto and colleagues who similarly compared oocyte retrieval, fertilization rate, and the number of top quality embryos, which they defined as an embryo with more than 7 blastomeres of equal size and less than 10% fragmentation. This study found that the number of top quality embryos was significantly lower in post-COVID individuals. Finally, Chamani and colleagues compared IVF outcomes of 1,881 women who went through the process between January and June 2022 to controls who went through IVF in 2019. The number of euploid embryos during the pandemic was significantly lower than pre-pandemic, and the number of blastocysts per patient increased. While fertilization rates of these eggs are unaffected, the quality of the preceding embryos appeared to be. While each of these studies have their limitations, mainly sample size, they do implicate COVID-19 as having a possible detrimental effect on fertility in terms of oocyte and embryo quality. Future work must be done to clarify contradictory results and track the long-term effects of fertility as one recovers from COVID and the antibodies associated with infection slowly fade. While what I have covered thus far is not exhaustive in terms of all the research done regarding COVID-19 and female fertility, it covers many of the latest findings. Beginning with the ovaries, reproductive hormones, and ovarian reserves largely appear to be unaffected by the SARS-CoV-2 virus and COVID-19. Oocytes, while expressing high levels of ACE2, are not found to have SARS-CoV-2 RNA, and neither does the surrounding follicular fluid. With that being said, SARS-CoV-2 antibodies are found in follicular fluid and have been linked to decreased vascular endothelial growth factor and interleukin-1 beta, providing the potential for decreased oocyte quality. When utilizing the opportunity to analyze oocytes during IVF egg retrieval, there is conflicting evidence on whether oocyte maturation and progression is hindered by COVID-19. However, the preceding embryos from COVID patients do demonstrate decreased quality in terms of euploidy, blastomeres, and embryo fragmentation. There is a lot of future work to be done. First, researchers can look to clarify the contradictory findings between research by adding additional controls and increasing sample sizes. Future work should also examine key reproductive hormone responses to COVID as they relate to areas outside of the ovaries. And finally, researchers should look to explore SARS-CoV-2's interaction with ACE2 and the associated balance of angiotensin systems to bridge that interaction with the actual presentation of fertility metrics mentioned above. And as COVID continues, we must examine longer-term impacts of COVID-19 exposure and fertility. We will next focus on the pregnancy aspect of fertility as it relates to COVID-19 exposure. It is no secret that certain groups of people are at an increased risk for acquiring this viral infection. Pregnant women have been constantly identified as one of these groups. They are less likely to experience some of the milder symptoms, but are more prone to failing respiratory and cardiovascular disease. As such, they are also more likely to be admitted to the ICU as compared to non-pregnant women of the same age. Since pregnant women are more likely to be vulnerable to this virus, it is imperative to understand if the virus could impact the developing fetus. To begin, we will watch this short video on the development of the placenta once a fertilized egg implants on the uterine wall.
Development of the placenta begins during implantation. When the blastocyst adheres to the endometrial epithelium, trophoblast cells differentiate into an inner cytotrophoblast layer and an outer syncytiotrophoblast layer. As the cytotrophoblast proliferates, newly formed cells migrate into the syncytiotrophoblast and lose their cell membranes, forming a rapidly growing multinucleated mass. The cytotrophoblast secretes proteolytic enzymes, and the syncytiotrophoblast sends out finger-like projections, allowing the blastocyst to embed into the endometrium. Lacunae, or spaces, begin to form within the syncytiotrophoblast. As the syncytiotrophoblast erodes the endometrial blood vessels and glands, the lacunae become filled with maternal blood and glandular secretions. Isolated lacunae fuse to form a network through which maternal blood flows, thus establishing early uteroplacental circulation. Toward the end of the second week, small projections of the cytotrophoblast begin to expand into the syncytiotrophoblast, forming primary chorionic villi. Early in the third week, extraembryonic mesoderm grows into these villi, forming a core of loose connective tissue, at which point the structures are known as secondary chorionic villi. By the end of the third week, embryonic blood vessels have begun to form in the extraembryonic mesoderm of the secondary chorionic villi, transforming them into tertiary chorionic villi. Cytotrophoblast cells from the tertiary villi grow toward the decidua basalis and spread across it to form a cytotrophoblastic shell. Villi that are connected to the decidua basalis through the cytotrophoblastic shell are called anchoring villi. Villi growing from the sides of the stem villi are called branch villi. These branch villi are surrounded by the intervillous space and will serve as the main sites of exchange between mother and fetus. By the fourth week, fetal blood flow is established. Poorly oxygenated blood is transported to the placenta from the fetus via paired umbilical arteries. These arteries send branches into the chorionic villi and divide into capillary networks at the terminal ends. Carbon dioxide and waste products are removed from the fetal circulation across the placental membrane to the maternal blood in the intervillous space. Oxygen and nutrients are transported across the placental membrane from the maternal blood to the fetal capillaries. Oxygenated fetal blood travels back through veins, converging to form a single umbilical vein that goes to the fetus. Blood returns to the maternal circulation through endometrial veins, forced out by the pressure of incoming arterial blood. The placenta nourishes the fetus throughout development and is delivered shortly after parturition. Development of the placenta begins during implantation.
During pregnancy, the placenta arguably plays the most important role in the development of the fetus as it provides oxygen and nutrients and removes waste from the baby. Thus, much work has been dedicated to understanding COVID-19's effect on the formation of the functional placenta. ACE2, the entry receptor for SARS-CoV-2, and TMPRSS2, the receptor that primes the virus for entry, are widely expressed in the human placenta. According to gene databases, the expression of ACE2 in the placenta is greater than that in the lungs. As the video detailed, the functional units of the placenta are the placenta villi and include syncytiotrophoblasts, cytotrophoblasts, and vascular smooth muscle of primary and secondary villi, all which express ACE2. In the maternal stroma, ACE2 is expressed in the invading and intravascular trophoblasts and in decidual cells. In the umbilical cord, ACE2 is found in arterial and venous endothelium and smooth muscle. ACE2 expression also appears to be phase-dependent, as it reaches its highest levels in early gestation, while in late gestation it decreases but is also found in the amniotic and yolk sac epithelium. With such high prevalence of ACE2 in the placenta, it suggests high potential for the SARS-CoV-2 virus to influence the proper development and functioning of the placenta throughout pregnancy. As we previously discussed, ACE2 plays a critical role in mediating the balance between angiotensins, specifically converting angiotensin 2 to angiotensin 1,7. This finely tuned system controls a number of functions, but plays a critical role in blood pressure and hydrosalinity of pregnant women. In terms of fetal development, this system has also been shown to regulate many aspects. The first concern would be its role in the regulation of maternal fetal blood flow during pregnancies. Angiotensin 2 also stimulates the trophoblast invasion, while ACE2 acts as a local autocrine and paracrine regulator during early and late pregnancy events. Pregnancy studies using ACE2 knockout mice result in increased angiotensin 2 and decreased angiotensin 1,7 levels, accompanied by abnormal placental functions and fetal growth retardation. With the SARS-CoV-2 binding to ACE2 and resulting in a downregulation of this receptor, literature suggests that this system could be detrimentally impacted and thus lead to placental and pregnancy complications. Moving on to if SARS-CoV-2 actually can invade these areas during the pregnancy, there has been extensive research primarily concerned with if a mother's infection can vertically transmit to the child. Hosier and colleagues analyzed the fetus of a woman with COVID-19 and found the prevalence of the virus on multiple facets of the maternal-fetal interface. They found evidence of the protein in both the placenta and umbilical cord, or evidence of the virus in both the placental and umbilical cord. Regarding the placenta, they found that the virus infected syncytial trophoblasts at the highest rate. Recall that we just discussed that these cells highly express the ACE2 receptor. Because these cells of the placenta are responsible for the circulation of nutrients and oxygen between mother and fetus, it raises the possibility of facilitating transmission to the fetus and also complications to the pregnancy. However, in the study, Hosier and colleagues found no evidence of fetal SARS-CoV-2 infection. However, contrary to the results of Hosier and colleagues, who examined just one woman and fetus, in many instances, there have been SARS-CoV-2 infected deliveries, suggesting that the vertical transmission is very much possible. Studies analyzing the placentals of infected deliveries have identified the SARS-CoV-2 spike protein in nearly all components of the maternal-fetal interface, including trophoblasts, decidual stromal cells, immune cells, epithelial cells of the placenta and amnion, and endothelial cells of the umbilical cord. Numerous studies reach different findings on how infected deliveries present, with some studies finding many new babies presented mild to moderate symptoms with shortness of breath, fever, pneumonia, and gastrointestinal symptoms, while other studies claim newborns tend to be asymptomatic. There has been a growing amount of research looking at what the implications are for newborns who are born with COVID-19. Here I highlight a few studies linking COVID at birth with detrimental effects. Knight and colleagues indicate that the virus influenced the development of neonatal nervous system and led to various neuronal dysfunctions and neonatal encephalopathy, along with other mental health conditions. Dahl and colleagues found newborns who are born with COVID as having liver abnormalities after being infected. He and colleagues indicate that SARS-CoV-2 in the third trimester of pregnancy could cause fetal kidney developmental injuries. And finally, multiple studies have implicated SARS-CoV-2 infected newborns as tending to have faster heart rates, tachycardia, or heart abnormalities. Future research into this topic is most definitely needed, especially tracking how infants exposed to COVID-19 develop later in life. While the implications of COVID-19 on the developing fetus and newborns can be difficult to study due to the semi-invasive nature of the work, studying pregnancy outcomes of women who have had COVID-19 while pregnant is a much easier area of explanation. The following information comes from a systematic review and meta-analysis which covered 42 studies involving over 430,000 pregnant women and comparing pregnancy outcomes. It is found that COVID-19 in pregnancy is associated with preeclampsia, a pregnancy complication characterized by high blood pressure and signs of damage to other organ systems, most often the liver and kidneys. Additionally, COVID-19 resulted in increased stillbirths or loss of a baby before delivering delivery, typically defined as a loss after 20 weeks of pregnancy. And finally, COVID-19 was associated with an increase of preterm birth before 37 weeks of pregnancy. Next, when comparing symptomatic versus asymptomatic pregnancies with COVID-19, we continue to see an even greater chance of preterm birth, as well as increased risk to have to undergo cesarean delivery. When looking at those who do not present symptoms, severe COVID-19 was strongly associated with preeclampsia, gestational diabetes, preterm birth, and low birth rate. So to ultimately review COVID-19 in pregnancies, women who are pregnant are considered to be a vulnerable group due to their increased risk of failing respiratory or cardiovascular systems. The developing placenta and other aspects of the maternal-fetal interface express the ACE2 receptor and thus may be capable of becoming infected. Additionally, the balance of angiotensins allow for the proper blood flow between mother and fetus. Studies find that in some cases, nearly every component of this interface can be found with the SARS-CoV-2 spike protein, especially the syncytial trophoblasts. Infection of these critical components can lead to vertical transmission of the SARS-CoV-2 virus, resulting in newborns testing positive for COVID when born. Such neonates may be at risk for neural, liver, kidney, and heart abnormalities, especially when the mother presents severe symptoms. In terms of pregnancy outcomes, the mother contracting COVID-19 while pregnant results in an increased risk of preeclampsia, stillbirth, and preterm birth. As these children develop, close attention should be given to how these complications proceed and if any others arise. Additionally, research should look into why some neonates are diagnosed with COVID-19 preceding the mother having it, while others are not. And that concludes my presentation on COVID-19's impact on female fertility. While I highlight some of the most relevant and commonly cited research, I once again acknowledge that this presentation does not begin to touch the tip of the extensive research that has and continues to be focused on this topic. For example, there is extensive research into the immune system response to SARS-CoV-2 and impacts on pregnancy in terms of inflammation and antibodies. Additionally, stress is known to decrease fertility, and it is no secret that the COVID-19 pandemic has added stress in some way to nearly every life. With that being said, I want to thank you for watching this video today, and please feel free to email me with any questions or comments regarding this presentation. Thank you.